All right, so we've talked about carbs, we've talked about fats. We're going to talk about proteins. Now, proteins are so important, not so much for their energy, but more so for the production of different types of proteins that in your body that you need. Specifically, I'm talking about enzymes. Okay, so but there are other structural features of like nail and hair and that sort of stuff that requires protein as well. Now, it can be a source of energy, but it's a last resort, as I mentioned, because if you're starting to use protein for your energy source, it's essentially, your body has essentially depleted the carbs, also depleted the fats, and now is actually eating, it's like breaking down its own muscle to obtain protein to then use that as an energy source. So it's really bad by the time you get to the point where you're using proteins, okay? Now, going back to enzymes, we'll learn about enzymes a little bit later in Unit 4, but just to preface it, like enzymes, they speed up the rate of reaction. They kind of act like a catalyst, but they're biological, so we call them, biological catalysts. Now, if we didn't have enzymes, the reactions in our body would be so slow that we'd actually die. That's how important it is to have proteins, because we need the enzymes. Breaking down the proteins into their individual monomers, so before we talked about carbs, they're monosaccharides. For proteins, we call them amino acids. I'm sure you've heard the term amino acids floating around, especially if you go to like the gym, you know? So what do they look like? The basic structure of an amino acid is the same, at least for your study design, because you're only asked to learn about alpha amino acids, okay? And it looks like this. You've got an amino group, you've got a carboxyl group, you'll have a hydrogen, and you'll have what we call a side chain. Look at it in a different way, right? Amino group, carboxyl group, hydrogen, and R. This is the side group. Now, you think to yourself, hang on a second, like the amino acids practically look the same. They kind of do, except the only thing that separates one amino acid from another amino acid is the differences in the side group, right? Which we'll explore a little bit later on, okay? So for your study design, you're only looking at alpha amino acids, which means that there's only one carbon in between the nitrogen and the carbon from the amine group, or amino group, sorry, and the carboxyl group. It's only separated by one carbon, okay? Now, you need to understand this structure because it's really important, and you'll come to see that intermolecular bonding is also going to be playing a part in this. Okay, making proteins. So what you're going to do here is these amino acids will come together by doing this. The carboxyl group of one amino acid is going to interact with the amino group with another amino acid. Now, they'll connect and form what we call a peptide bond. Actually, it's not a peptide bond, but hey, the peptide bond is technically that bond between the carbon and the nitrogen. Biologists will tell you it's a peptide bond, but hey. So the whole thing, like that, is an amide functional group. We call it an amide functional group. That's what we look at it from a chemistry perspective. And it's the amide functional group that links them together. So, like the fats, it's the ester functional group that holds together the glycerol and the fatty acids. In the carbs, it's the glycosidic link that looks like this, that holds together one monomer with another monomer. Okay? So if you have a look at the animation, I like these animations, you'll see that, again, water is lost while you form this amide functional group. Again, because water is lost, we call it a condensation reaction. Okay, so the COO interacts with NH2, and that forms the amide functional group, which is pretty cool. So you can imagine that even though, say for example here, this amino group's reacted with this carboxyl group, there's a carboxyl group here that's exposed that can then interact with another amino group, and the process continues. So you got one amino acid, another amino acid, dipeptide. You've got 3, tripeptide. Then you've got polypeptide. Okay, so you use a long chain of amino acids to make your protein. Like the fatty acids, you don't have to memorize any of the amino acids because they have all of the amino acids that you need in your data book. Now, I want to show you the structure. NH2COOH, NH2COOH, hydrogen, hydrogen. Can you see that? So the structure is common to all of these amino acids, right? However, you'll see that they'll have different side groups, different side groups, different side groups or side chains. That is actually what dictates the behavior of each of these different amino acids, whether they're soluble, whether they're not soluble, whether they interact with, I don't know, like how they also interact with each other in a structural formation, okay? So, when we're talking about structure, there's 3 main structures that I need you to know. The primary, the secondary, and the tertiary. The tertiary version is actually the functional version of the protein. So if we're talking about enzymes, it's at minimum at a tertiary level. It's a 3D structure, okay? Now, they all have different shapes, and it's affected by the types of bonds that are actually holding them together. So if we have a look at this, right, this is the different structures. Okay, starting with primary. Primary is just when all of the amino acids link together in a chain. I want you to think of it like a bracelet, right? It's just a simple bracelet, beaded bracelet. And so it's connecting by those amide functional groups, yeah? Those COOH and the NH2 come together, and then you're stuck together in like a bead chain, right? Now this occurs by covalent bonding, so the amino acids are connected together. Once you've got that nice long chain, what's going to happen is, depending on the bonds and where the bonds will occur, it's either going to take one of two forms. You're going to have an alpha helix or a beta pleated sheet, right? But regardless of which structure, they're both held together by hydrogen bonds. It just depends on the location of the hydrogen bond. Now that hydrogen bond will form between the O of the COO of one amino acid and the hydrogen of an NH of another amino acid about 4 positions away. So if that's the case, you're going to give yourself an alpha helix, that coil. If it's slightly different, like different segments, again, still hydrogen bonding, you're going to get a beta pleated sheet, which is like, imagine like corrugated iron on your roof, or just like, you know, if you take a long piece of paper and you start to fold it and it comes out like an accordion kind of thing, that's the beta pleated sheet. So you've got those two structures. So you've got your primary, long string. That long string then starts to interact with each other using hydrogen bonds. It's either going to be a helix or it's going to be a beta pleated sheet, depending on where the hydrogen bonds are occurring. You get to the 3D structure, it's different again, but the 3D structure depends on the bonds between the side chains. Okay, this occurs between the R groups. That's why I was telling you it was so important with the amino acids. Those side groups, the differences in that will dictate the types of intermolecular bonding. So it might be hydrogen bonding. It might be disulfide bridges, right? But that's only for the amino acid that has the SH on the side group, and you'll see it in your data book. It could be dispersion forces. It could be ionic bonds. I don't know. I'm just listing them all here for you to think about, because you have to be able to look at the structure of the amino acid, look at the side group and go, oh, it's this one because the side group is completely non-polar, or it could be dipole-dipole interactions because the side group is polar. I don't know. I really don't. But you just need to know that that's how those bonds form. It's between the side groups and no longer between, like, you know, coiling or beta pleated sheet. Quaternary structure is like taking the 3D structures and putting those 3D structures together, okay? And an example of this is like hemoglobin in our body. Now, hemoglobin is like the 4D structure, the quaternary structure, made up of like the globulins, right? 4 of them. So again, same thing. Multiple polypeptide chains that can be held together by any of the bonds from above, right? Ionic dispersion, disulfide, hydrogen bonding, like you name it. It just depends on what groups are present and therefore what types of bonds can form. Okay, so to recap that, primary, linear beads, right? Held together by those covalent bonds, which is through something that looks like this, all right? So the rest of the one amino acid, this is the rest of another amino acid. Secondary are the alpha helix or beta pleated sheets, and that will be due to the hydrogen bonds that occur between the CH and the NH, right? It's hydrogen bonding. The tertiary, all sorts of different types of bonds. It could be anything, any of the options that you actually know about. We actually even, I didn't even mention there's another one called an iron dipole. Okay, that's also possible if you've got a side group that's like can form an ion, the other one that can form a dipole. Because they're oppositely charged, you just come together, right? It just depends on the side group. It just depends on the side group. So I can't tell you what it's going to be. This is why I keep stressing it's so important for you to be able to pick out intermolecular bonds, because it shows up so much everywhere. Quaternary, same types of bonds. Again, it just depends on what's available. Another look at them. So I keep telling you that it's for tertiary structures, it's the hydrogen bonding between the NH and the COO. I just wanted you to see it for yourself. Okay, here, this interaction here. But same thing here, NH and the COO, this bond here is a hydrogen bond. Okay. Now, when it comes to, say, a tertiary structure, now each of these shown are, side groups. So this is a side group of 1, so you can see it's an ion. And here, that's another ion, so it's ionic bonding. Here, this is an example of just dispersion. Why? Because each of these side groups are only nonpolar. And for nonpolar, we know that it's only dispersion, and so on and so forth. So you see where I'm trying to get at with this, right? Okay. If it's condensation to make them, it's hydrolysis to break them. So you can see now it doesn't matter what type of biomolecule you're talking about, carbs, fats, or proteins, it's always going to be condensation to make a bigger version, hydrolysis to break them down. Okay, so put the water back in, give it back to where it was lost from, and then you have your separate amino acid. So that's an amino acid, that's another amino acid. Okay. So we spoke of, like, this is all in the context of energy, but to be honest with you, the way that I've broken down these biomolecules is in more detail than you need to know right now in Unit 3, Area Study 1. In fact, I took some of the stuff from Unit 4 just because I really found that there's no point kind of leaving you halfway here and then going back later. You might as well learn it now so that you can relearn it later, right? So it's already floating there in your brain. So in the context of energy, this is in your data book, so you don't need to know. But as I mentioned, fats and oils are the best in terms of, if you're just talking about straight up energy, it gives you 37 kilojoules per 1 gram that you consume. Versus proteins, which is 17, and the carbs, which is 16. The funny thing is we predominantly use carbs as our energy source first, right? It's easiest to break down. It gives us less energy. Proteins, we don't want to because we don't want to start breaking down tissue from our body. But fats, like, they're the highest energy, but it's kind of used second. Carbs will go first, then fats, because it actually takes a little bit longer and it's more energy consuming in our body. But yeah, so you'll need to be able to do any calculations. So if I say, hey, I've got 2 grams of fats, you're going to say, yep, that's going to be 74 kilojoules of energy. That's all. And I know you know how to do basic math, so you're all good. So summary, doesn't matter what biomolecule, condensation to make them, hydrolysis to break them. Carbs, held together by glycosidic bonds. Okay. Lipids, ester functional groups. So it's this COO between the fatty acid and the glycerol. Okay. Proteins, peptide bonds. I want you to call them amide functional groups though, right? And that looks like this, right? And in order to break them back into their individual amino acids, again, just put water back in. Cool. All right. Thank you so much for hanging out with me. I hope that was helpful. Always remember, no matter what anybody says, yeah, you can.